The paper presents a novel deep learning model, White Matter Geometry-guided Diffusion, WMG-Diff, for imputing tissue microstructure in diffusion magnetic resonance imaging, dMRI, tractography fiber clusters. The model utilizes a score-based guided diffusion model to address missing data values due to variability in scan quality, interindividual anatomical variability, and other factors. The WMG-Diff model consists of three main components: a deep score-based guided diffusion model for imputation, a white matter atlas geometric relationship guided denoising function to guide the reverse denoising process at the subject-specific level, and training and evaluation on a large dataset with 9,342 subjects. The authors propose the use of score-based diffusion models, which have been successful in various deep generative modeling tasks, and extend this model by leveraging information about white matter fiber cluster geometry to define a guided denoising function. This novel approach replaces a random data reverse diffusion process with a geometry-guided reverse diffusion process, improving performance on downstream tasks such as non-imaging phenotype prediction. Comprehensive experiments demonstrate that the proposed WMG-Diff model outperforms state-of-the-art methods for tissue microstructure imputation and phenotype prediction. This work marks an advance in leveraging diffusion models to analyze microstructural properties and addresses the problem of missing data in large brain datasets, providing anatomical features for disease prediction, anatomical tract segmentation, surgical brain mapping, and non-imaging phenotype classifications. The proposed WMG-Diff model utilizes a deep generative approach to impute missing diffusion MRI microstructure data, leveraging anatomical relationships within white matter geometry. This is the first work to explore dMRI microstructure imputation and apply generative diffusion models to this task. The model focuses on fiber clusters geometrically similar to those missing in the dataset, hypothesizing that this will guide the reverse diffusion process to impute more accurate tissue microstructure values. The dataset used for evaluation is the large-scale, multi-site adolescent brain cognitive development (ABCD) dataset, which includes 9,342 young children aged between 9 and 11 years. To standardize the dMRI data across 21 different acquisition sites, the data was harmonized to remove scanner-specific biases while preserving inter-subject biological variability. The fiber cluster parcellation employed was created to be consistent across subjects, allowing for the calculation of FA values and identification of missing values. The proposed WMG-Diff pipeline imputes missing FA data for input to a downstream sex prediction testbed task. The approach utilizes information from anatomically similar fiber clusters to aid the reverse diffusion generation process, leveraging the geometric relationships between WM tractography fiber clusters. The model's performance is evaluated on a harmonized, large-scale dataset, showing that WMG-Diff outperforms popular state-of-the-art imputation methods. In summary, the WMG-Diff model introduces a novel deep generative approach for imputing missing dMRI microstructure data, utilizing anatomical relationships within white matter geometry to guide the reverse diffusion process. The model's performance is evaluated on a large-scale dataset, demonstrating its potential in improving the accuracy of tissue microstructure values for various applications in neuroscience research. The proposed method leverages a geometry-guided diffusion model to address missing FA data in tractography analysis. It utilizes the minimum average direct flip pairwise streamline distance between fiber clusters in the org atlas to establish geometric similarity. This similarity is crucial for guiding the training process of the diffusion model. The diffusion model is a score-based conditional model, which is effective at learning conditional probability distributions. It comprises convolutional layers, a diffusion embedding module, transformer encoders, and residual blocks. The training dataset consists of actual missing FA data, non-missing training data, and synthetic missing data generated by randomly dividing the non-missing data into guided observable values and imputation targets. The motivation behind choosing this model is to impute missing FA values considering the subject-specific tissue microstructure distribution. A geometric relationship-guided denoising function is proposed, focusing on frequently missing FA data due to anatomical or scanned protocol variability. Guided observable values are selected based on geometric distances calculated from the White Matter Atlas, aiming to leverage information about WM anatomy and the distribution of missing fiber clusters to guide the training process. The architecture of the WMG-Diff guided process involves splitting non-missing data into observable values and imputation targets using geometric relationships. At each time step, the imputation target undergoes a forward process to add noise until it becomes fully noisy. The reverse diffuse process leverages guided observable data at each time step to denoise the noisy target and minimize the difference between the noise and the output. This approach aims to provide accurate imputation of missing FA data in tractography analysis. The research paper discusses the development of a white matter geometry-guided diffusion model for imputation, focusing on its application in a downstream sex prediction task. The model is trained and evaluated using a diffusion process with specific parameters and is compared against state-of-the-art methods and common neuroimaging data imputation practices. The results demonstrate the effectiveness of the proposed WMG-Diff model in terms of root mean squared error (RMSE) and accuracy (ACC) metrics, outperforming existing methods. An ablation study also highlights the importance of the geometry-guided denoising function in achieving better performance. The model's performance is evaluated on an NVIDIA RTX A5000 using PyTorch version 1.13, and the results are quantified using RMSE and ACC metrics. The study contributes to the field by providing a novel approach for imputation in neuroimaging analysis, particularly in sex prediction tasks. Here is a combined summary of pages 9 and 10. The proposed white matter geometry-guided diffusion (WMG-Diff) model is a conditional score-based diffusion-based approach that leverages prior knowledge of white matter structure geometric relationships to impute missing fractional anisotropy (FA) values. The guided denoising function enhances learning during the reverse diffusion process. Evaluations on the ABCD dataset demonstrate that WMG-Diff outperforms widely applied state-of-the-art methods. To investigate the effectiveness of data imputation, a logistic regression approach is employed to predict sex based on the imputed FA values. Results show that WMG-Diff outperforms widely applied imputation methods, and sex prediction accuracy using WMG-Diff imputation is almost identical to using the full dataset. The success of the proposed imputation method suggests that WMG-Diff can generate accurate tissue microstructure data, which benefits downstream tractography-related prediction tasks. This study uses public ABCD imaging data, and approval was granted by the BWHIRB for the use of the public data. The study is supported by several National Institutes of Health (NIH) grants, the National Key R&D Program of China, the National Natural Science Foundation of China, and the University of Sydney International Scholarship and Postgraduate Research Support Scheme. The referenced papers provide additional context and supporting information for the development and evaluation of WMG-Diff. These papers cover topics such as multiple imputation by chained equations, the adolescent brain cognitive development study, harmonized diffusion MRI data, and geometric deep learning frameworks for pointwise analysis of tract microstructure. The research focuses on developing novel diffusion models for imputation in various fields, including brain imaging and connectomics. It leverages white matter geometry to guide diffusion models for predicting neurocognitive measures and language performance. The methods employ supervised contrastive learning and graph neural networks to impute missing data in connectome-based predictive modeling. The results show improved accuracy in predicting non-imaging phenotypes and language performance cognitive scores. Additionally, the models demonstrate test-retest reproducibility of white matter parcellation and can be used for consistent white matter tract parcellation across the lifespan. The study highlights the potential of diffusion models in enhancing our understanding of brain connectivity and its relationship with cognitive functions.